I feel very honored, and it's certainly a great pleasure to be here to speak to you from an industry perspective. I thank Mihai and Joachim Schulze for inviting me here and to give the opportunity not only to introduce what we're doing with the Human Functional Genome Project, but also how industry more in general looks at what you're doing and how that may play out in the future. Now, let me please start off and allow me to start with a personal note. I am a treating physician. I do treat HIV patients since 25 years. I shifted gears 4 years ago when I went to the dark side of the moon. But there are some bright spots there, so this is where I am. This is ViiV Healthcare, and the specialty of ViiV is we are focusing exclusively on HIV. We're a joint venture of GlaxoSmithKline, which is our major shareholder, and a little bit Pfizer, and later Shionogi joined in, and we are just focusing on HIV, so focus, focus, focus. And we are trying to actually bring in big minds and the brightest people from academia to join industry in this kind of biotech startup, which is embedded into a larger framework of big pharma. Now, having said that, what I'm trying to do now in the next 10 minutes is why are we interested in a cross-omics public-private partnership like with the Radboud University, and how may this fit into the lifetime strategy? So let me start off by saying, that we can really treat HIV very well today, but there is no cure yet. And that comes with a lot of challenges with regard to lifetime therapy, cumulative toxicities, eventually development of resistance, but also cost and access is an issue. So we believe that a cure is mandatory and to keep on working on that, and cure is on the continuum of treatment. So if a patient comes to me today and I have to tell him that he or she is infected with HIV, we do have very many good options to actually treat patients. And basically, you can take one pill once daily containing only 2 drugs today in combination to really make a very successful and very suppressive therapy for a lifetime. You can even have perhaps tomorrow quarterly injections every month or every second month. We have actually within our 9-year history at ViiV, we have launched 5 products into the market against HIV. We have just filed the 6th one, which is an injectable, can be given every month, and we are aiming at another submission and filing next year. So this is a unprecedented success story. Why are we so successful? Because we're nimble and we're focused. And what is actually our ultimate goal is to cure HIV. But there are many barriers to HIV cure, and multiple barriers are just a few of them listed here. One, which Mihai has already alluded to, is the latently infected cell pool, or the so-called viral reservoir. You do have, and there's a lot of debate about that, subclinical level of viral replication. You do have anatomical sites which may not be approached by each drug similarly effectively, and you do have persisting immune dysfunction. Now, HIV disease persistence is largely driven by viral reservoirs. So our current therapies suppress successfully the production of viral RNA in plasma, but it does not have any good effect on the viral reservoir, namely the DNA, which is integrated into latently infected cells, do not respond very well. And here, this viral persistence is actually associated with chronic inflammation, and this chronic inflammation, we do believe, is actually driving a large part, at least in a good number of patients, what we call non-communicable diseases. So that is what we call immune aging. And these are the effects on the cardiovascular system, the endothelial dysfunction, dyslipidemia. NASH and metabolic syndrome is a big clinical problem, as well as end-stage renal failure, and also metabolic diseases like frank diabetes to develop osteoporosis, etc., etc. So our patients with HIV are successfully treated in terms of viral replication, but there is some immune activation persisting, which we believe is due to the fact there is viral persistence and release of viral proteins, okay? So HIV hides in long-lived cells and also in tissues. And just to set the frame here, it is that latently infected cells, which are here in green, retroviruses integrate into the genome, and then, once it is integrated and the cell divides, it will pass on to the daughter cells, of course. So this is what we call homeostatic proliferation. So even in the absence of active viral replication, just by division of infected cells, the reservoir will be replenished on and on. And it has been a long-standing dogma over the past decades that retroviruses, as HIV, will integrate randomly. This is not true, and there is one of the champions of this integration site analysis here in the audience, Moncef Benkiran, showing that the fate of a cell and the cell activation pattern of a cell and the genetic pathways which are turned on when the cell gets infected will pretty much determine where these retrovirus will integrate into the genome. So this is a strong link to the cell level which you are studying. Other problems which we are facing is that what I call the macro compartments. So there are different cell types which are infected. Usually it is CD4 positive T lymphocytes, and the latent reservoir made in the great majority consists of naive T cells. But we do also know that in the brain microglia are infected and that there are T follicular helper cells in the lymph nodes play a special role, gut-associated lymphoid tissues play a role, etc., and the genital tract, of course. So we do also have macro compartments which are playing a role. So what we do have very well established is depicted here on the left side. We can treat, hopefully early as diagnosed, restore immune function, prevent AIDS, and improve the life of our patients. And clearly, in the vast majority of patients who are diagnosed early and treated well, we can really treat them as good as reaching them a normal lifespan. But there is, on the host side, much to do. We do have persisting chronic activation and inflammation. Therapeutic options are needed which target now host responses and host factors to enable more efficient immune functions, also to control HIV infection in the future with less interventions and lower the reservoir, and eventually and ultimately hopefully allow us to reach a cure. So that is the first 5 minutes of my talk. The last 5 minutes I will spend on giving you an industry perspective in general. Big pharma investments in 2018 for the first time exceeded $100 billion in 1 year, okay? And you can see this is the annual growth rate, which is about 6%, and it increased from 2012 to 2018, as you can see, by 32%. So there is a steady increase, $100 billion investment each year, and a steady increase in these investments. Now that led to a record number of innovative medicines which were launched in 2018, bringing 59 new treatment options to patients. And here in the circles you can see the therapy areas. And what you can see is 27% come from oncology, and then followed by 20% infectious diseases. So oncology and infectious diseases and neurologic diseases are actually drivers here in this investment, okay? What you can also see is that half of those are in rare diseases, in so-called orphan drug designations, and we do see already 1/4 not being small molecules but being biologics now. A part of them are actually either stratified and diagnosed and with kind of predicting response by predictive biomarkers, and then you do have a number of other compounds as well. Now, new drugs launched in 2018 took an unacceptable 14 years on average from filing of the first patent to the launch of a product. 14 years. And this 14 years is not only hard work and taking much too long, but it's also very costly, okay? Now, what I've shown here is also that, there is a large variability. There are some drugs which have actually taken 30 years for tafenoquine, an antimalarial, to develop into the clinic from the first filing of the first patent. This is unacceptable. What is the contribution of biotechs? Concentrate on the right side here, please. In 2018, These are the new drugs which actually originated from small biotechs, which eventually were launched later. And at the originator, in 2/3 of those products were actually biotechs. And then they moved on to develop these drugs, in collaboration with Big Pharma, or Big Pharma bought the biotech, so that it turned out at filing that it was almost equal, from Big Pharma and the distribution and biotechs at the time of filing. The next generation biotherapeutics have more than doubled since 2015, and all of these different disease areas have actually contributed to that increase in biotherapeutics. But again, as you can see here in green, the darker green, infectious diseases do contribute also to this field of increasing biotherapeutics to develop. Now, the composite success rate of clinical development stages from phase 1 to launch actually fell to 11% only. What does it mean? If you develop 10 drugs or file 10 patients, only one will make it into the clinic. And what you can see here is the likelihood on the bottom here, the likelihood of failure in different stages, or here I shouldn't say failure, but success, so is the glass half full or half empty? In this sense, it is half full. And what you can see here is the attrition rate between phase 1 and 2 is the biggest, of course. But again, if we could only make it a from 10 to 20% success rate, for us as an industry, this would be an immense save. And it would be an immense return on investment if we could make this happen. So let me end up by giving you one of the perspectives. I'm now head of translational medicine at ViiV. I thought translational medicine means from bench to bedside, but also bringing the real problems from the bedside to the bench. But this is not my real remit. My real remit is actually to be translating language of academia into industry language and vice versa. Your language in academia, my language in academia, let me take one expression, currency. What is the currency you're paid in? Your currency is impact factors, successful grant applications, reputation, and maybe awards. Our currency we're talking about is return on investment, shareholder value, and basically increase in growth and sales. Okay, that's our currency. So here we need to find a real good partnership to increase these PPPs. We do believe that genetically validated target identification is crucial to identify as well better biomarker algorithms, either for the selection of the right patient population or also for allowing us to assess if our medicines are working well or not so well. We have to minimize by doing that to this attrition rate, which I have showed you. We have to create funds to help the transitioning from discovery to early development and to enable a higher success rate in early development stages. And we have to use, ultimately, innovative trial designs, ensure early alignment with regulatory bodies, of course, and account for moving towards from small molecules to biological and biotherapeutics, and ultimately transition to more precision and personalized medicines. And my last slide is some words of wisdom for success, and I have borrowed this slide from my colleague Ray Shinazi. Ray developed a couple of very successful drugs, including some drugs for HIV, but also for hepatitis C, one of them sofosbuvir, so he knows what he's talking about. And his words of wisdom and my words of wisdom is know what is wrong with your drug before someone else finds out. That's number one. Number two, if you're going to fail, fail fast. This means you have to aim high and fail cheap, all right? And then you are not a failure. It's the drug which is the failure. You have to be trying, keep hard, and work on. Doing nothing has consequences. Don't just sit and wait, do something. Make every experiment count towards an IND or NDA filing and put the patient's need at the center of what you're doing. This is ultimately what we need to really focus on. And this is, I believe, the most important message from me to you today. Choose your collaborators wisely. Having great collaborators who deliver is critical. Thank you very much.
Thank you very much. I particularly liked your term, you're successful treating patients through lifetime. Right?
Yeah.
Yeah, I mean, maybe I can ask for a general view on preclinical models. from the big pharma, do you think to, as you said, to stop fast, to fail faster would help us? Is much more needed on the preclinical level in your view? And if so, the question to Mihai and to both of you on the HIV, how much do we have any preclinical models that could help?
You want to start or me? Or?
Yeah, you have to come here. So there are some clinical models. Of course, simian models probably the best, some mouse models in humanized mice also. But I do think that actually human immunology in the case of HIV is by far number one. And of course, the experimental models could help for very specific questions.
I agree. It always depends on the question you have. So if you want to study toxicity, sometimes, or even pathophysiology, mouse models would be great. But if you ultimately would come in now with something which is immune response modifying, for instance, the mouse is completely different to the human immune system. And by the way, to infect the mouse, even a transgenic mouse which is repopulated with the human immune system is not meaning that this is a normal human immune system. So I doubt that this will be a real good model. Even what we have learned in the past, using simian models like macaques, chimpanzees almost never. The chimpanzee, by the way, is a very interesting model because HIV is the chimpanzee is the only natural host for HIV, but the chimpanzee doesn't get sick. So if we could only understand why, that would help us a lot. So there are some models, but I'm a big believer. Don't waste your time too much with monkey and with mouse models. Go into the humans, go to people like Mihai and his group and, try to find out what are the determinants in the human system and take it from there.
Okay. Thank you for the beautiful talks. I haven't heard anything about vaccine.
Yeah.
What you, Mihai, just presented, I think applying that to understanding, vaccine response, immune response to vaccine, I think would be just wonderful, because we know that we need vaccine for HIV, and if we achieve a good vaccine for HIV, I think we will be able to easily make vaccine against many other infectious diseases. What do you think?
Well, I cannot agree more. Let's put it very simple. And I
Yes.
think that understanding indeed variability in the immune responses in the human host, understanding why some people are, for example, resisting to the infection with HIV, why some of them are rapid progressors, and so on. And this is one of the aims of what, because of the time, I didn't have the time to present that, but this is also one of the aims that we have. Understanding that will greatly help us also in vaccine development, and actually, certainly, HIV, I do think that is one of the more difficult vaccines, so that would help also for the other infections.
Absolutely. Yeah, I couldn't agree more. And, as Mihai said, looking at the outliers of the Gaussian shape in this, human functional genomics project will help us to understand the correlates of immune protection, for instance. And this is really needed, and as well as me that there is a hype about BNAbs and using these broadly neutralizing antibodies as an epitope scaffolder and all that stuff, but I think what we got to do really is to really understand why these rare patient phenotypes do exist, and if we do come across why these rare phenotypes actually, and how these people and individuals make it happen, that they can control the virus without any intervention, I think we are a big step further.